Fluorescence-mediated tomography is a highly sensitive imaging technique to quantitatively assess the fluorescence distribution in anesthetized mice. Many targeted fluorescent probes are available, which allow imaging of angiogenesis, apoptosis, inflammation, and others. In this movie, we will show how hybrid imaging with FMT and µCT is performed at our institute in Aachen. Chapter 1. The devices. This is our FMT. FMT stands for Fluorescence Molecular Tomography. Tomography means that 3D images are generated. The FMT is highly sensitive for imaging near-infrared fluorescence in mice. Now the front panel is opened to show the inside of the FMT. At the bottom is a laser mounted on a 2D servo. The laser emits light into the mouse, which is held in a partially transparent mousebed. Above the mousebed is an array of LEDs as an alternative light source, which allows the FMT to operate as a regular reflectance imaging device. A filter wheel is mounted below the lens, which captures the light for the detector on top of the device. This is our µCT. The µCT is equipped with 2 X-ray tubes and 2 flat panel detectors, which allow acquisition of dual energy scans. Chapter 2. µCT-FMT Scanning Protocol. Both FMT and µCT are built to image mice. Before imaging, mice are anesthetized with isoflurane. For the FMT scanning, the hair of the mouse needs to be removed, which works well with dehairing cream. Some strains of mice can develop rashes from the dehairing cream. Therefore, it is recommended to monitor the mice for skin changes and to contact the veterinary staff for care if needed. Also test the tolerance on a small batch of any new strains of mice to start with. To avoid hypothermia, the mouse is placed on a heating pad. To inject contrast agent, a catheter is placed into the tail vein. Due to the small size, this is quite difficult and requires some experience. If blood flows back into the catheter, it is placed properly. Through the catheter, fluorescent contrast agents and CT contrast agents can be injected. To avoid volume overloading, at most 5 ml per kilogram body weight should be injected, which means 150 microliter for a 30-gram mouse. For scanning, the mouse is placed inside a multimodal mousebed. The mouse may have some symbols drawn onto the tail for identification. It is important to avoid such things on the torso, as it may impact the optical scanning. The mousebed is closed and the depth adjusted to tightly hold the mouse in a fixed position. Be careful and monitor the breathing, because tightening the bed too much can suffocate the mouse. Nude mice are commonly used for oncologic research due to their immune suppression. The fact that they are nude is a fortunate side effect of the genetic mutation. Therefore, the laborious hair removal procedure can be omitted. It should be checked that the mouse breathes properly. And, if necessary, the mousebed should be adjusted accordingly. Then the mousebed is placed inside the µCT. The tubes, which transport the isoflurane gas, are switched to maintain the gas flow inside the device. Then the µCT is closed to enable the X-ray shielding. The µCT will only start scanning if the lid is closed. Using the buttons on the µCT, the mouse can be driven into the µCT. At the µCT control computer, a topogram is acquired and shown. The windowing settings are adjusted to better see the mouse. 1 or more subscans can be placed. Their position is indicated by the light blue regions. Usually 1 to 3 subscans are sufficient. After starting the scanning, the progress is shown with dark blue progress bars. Our flat panel µCT scans the subscans subsequently, which is different from a clinical spiral CT. Using the buttons, the mousebed is moved to the front again. The shielding lid is opened and the anesthetic tubes are switched. The holder is carefully removed from the mousebed and the anesthetic tube is pulled out. This is necessary because the anesthetization in the FMT does not depend on this small tube. Instead, a small chamber inside the FMT is flooded with anesthetic gas. Now the mouse bed with the mouse is brought over to the FMT and inserted. On the FMT control computer, the scanning field of view is adjusted as well as the sampling density. Usually around 120 points are used. The scan starts after pressing a button. The first pass of the FMT acquires a transillumination or excitation image for each laser source point. In this movie it is shown in fast forward mode. Usually it takes around 5 minutes. You can see that much less light passes through the upper body compared to the lower body. This is because the organs with higher relative blood volume, such as heart, liver and kidneys, are more in the upper body. Blood is the main absorber for near-infrared light. The second pass runs over the same source points with a different filter, which only lets the fluorescent light pass through. Chapter 3: Interactive Organ Segmentation. To fuse the data from both devices, markers are used, which are built into the mousebed. The markers are also visible in a reflection image acquired by the FMT. The markers are actually simple holes and do not need to be filled with any fluorescence or CT contrast agent. At our institute, we developed a software program that performs the marker detection and fusion automatically. The shape of the mouse, as well as heterogeneous absorption and scattering maps, are automatically estimated using the µCT data, as described in our recent theranostics publication. These parameters are important for quantitative fluorescence reconstruction. To measure the biodistribution of the fluorescence, an organ segmentation is needed. We generate such a segmentation interactively using a software named Immulytics Preclinical, developed at our institute. In the following, such a segmentation is shown in fast forward. An experienced person can perform it in around 10 to 20 minutes. First, the CT dataset is loaded. It can be inspected in 3D using an ISO surface rendering. By changing the windowing settings, the ISO value can be changed. For example, to visualize bones or the entire mouse body with the mousebed. Then the overlay is loaded. The signal for this example appears in the bladder. Now the overlay visualization is turned off to concentrate on the anatomical segmentation. In native µCT scans, the lung is easily found due to the strong negative contrast to the other soft tissue. The big structure inside the lung is the heart. Let's segment the lung. First, all voxels below a certain value are segmented using thresholding. This appears as green. The lung is a connected region which can be separated using a filling operation, similar to the paint bucket in a painting program. The trachea can be separated from the lung by cutting and filling. Convex organs, such as the bladder, can be segmented by drawing scribbles to delineate the borders of the organ. More scribbles can be added until a sufficient accuracy is reached. Non-convex organs, such as the intestine, can be segmented piece by piece. The liver appears as a homogeneous region and has a more complex structure since it consists of multiple lobes. The segmentation can be saved to disk and loaded into the program. Using such a segmentation, the fluorescence signal can be assigned to the organs. The program computes these amounts and saves them as an Excel sheet. For longitudinal scans, the segmentation needs to be performed for each time point again, because the intervals are usually too long to keep the mouse anesthetized in a fixed position. Therefore, the segmentation is a laborious task if many mice and many time points are involved. To quantify the results, load an overlay and a segmentation. Click Set Batch Settings to let the program remember the current settings. Now click Batch Statistics to tell the program to compute the values for all regions in all µCT-FMT scans. This will take a few seconds. Then the statistics are saved in one spreadsheet file. This is convenient because the user does not have to merge dozens of files himself. Based on this file, the organ curves can be computed. Chapter 4. Representative results. To test that the fusion works properly, we used an agarose phantom. Some titanium oxide powder was added for scattering. To realize an irregular shape, we cut away some parts. Several small inclusions filled with fluorescence and CT contrast agent were built into the phantom. Since the FMT does not know the true shape of the object and assumes a simplified shape, the reconstruction is not accurate for objects with irregular shapes. Therefore, we implemented another reconstruction that uses the shape derived from the µCT data. As you can see, the signal localization in the phantom is much better. To inspect the in vivo data, let's go through the imaging time points. This is the pre-scan. What we see is basically just noise and artifacts. If we go to the next time point, the one after injection, we see much more signal, but the windowing settings are much too hard. Using the windowing dialog, you can adjust the windowing settings. We see most of the signal in the bladder. If we go to the next time point, 2 hours after injection, we see some signal outside the mouse. This is because the mouse urinated the fluorescence onto the mouse bed. By further adjusting the windowing, we can see some signal at the spine and knees. Now let's go to time point 4 hours after injection. The signal from urine is gone now, and we see fluorescence at the spine and the knees. For the next time points, 6 hours and 24 hours after injection, we see the same. Now let's go over to the next mouse. The pre-scan shows nothing, using these windowing settings. The scan 15 minutes after injection shows strong bladder signal, and so on. In this study, we found high concentrations in the urinary bladder shortly after injection, as a consequence of the fast renal excretion of this probe. Furthermore, the signal in the spine quickly rises and remains relatively stable throughout the later imaging time points. Chapter 5. Conclusion. In conclusion, we show a multimodal imaging protocol to combine the strengths of fluorescence molecular tomography and microcomputed tomography. The anatomical µCT data enables an improved fluorescence reconstruction by using the shape of the mouse. Furthermore, it is useful to generate organ segmentations which are needed to extract quantitative measurements from the image data.